everybody. I think we can get started since it's about 2 p.m. Thank you everyone for taking the time to learn about our COVID-19 competitive landscape map today. We hope that this will help you in sort of your respective positions to navigate really what's going on from the pandemic response standpoint. Just a few housekeeping items. Please use the question and answer button below on the Zoom taskbar to ask any questions that you have throughout the tour. We'll try to address as many as we can. There's an upvote button, so if you see a question that's particularly relevant to you, please feel free to upvote it and we'll try to prioritize those. We'll also try to keep the presentation open for a little while after the event ends so that you can review those answers. Please note that the presentation should be taken as off the record. If you would like any quotes pertaining to the presentation, please reach out and we can supply them. And lastly, we would be really happy to engage with you further after this map tour if you are sort of working on the pandemic response and would like to work as thought partners together. So please do reach out if that's of interest to you. And we'll be having a few more map tours as the weeks go by to kind of help, show how the story is evolving over time. So please feel free to join those. So with that, I will hand it off to Peter Kolchinsky, who's the founder and managing director here at RA, to kick things off.
Thank you very much, Alyssa. So welcome everybody. Some of you may be coming back. This may be the first for everybody, for some folks. I would point out that we've started to record these Zoom tours and post them to our website. And that allows us to actually evolve them. And what we will encourage people to do is for certain kinds of foundational information, like the kind of introduction that we gave at the last Zoom tour, we'll end up directing people to review a prior Zoom tour so that we can start to build on these over time. What we'd like to do is to start using these tours to teach something new and go deeper into our thinking about the COVID response. So I'm gonna share my screen. And before I jump into the Zoom tour, I'm going to share with you what we think is the, apologies, I'm struggling here to, there we go. Okay, share. What we think is the objective of the companies that are developing vaccines. The objective everybody knows about is to develop vaccines that are going to allow us to end the COVID pandemic. So something that's good enough, that's protective. But there's another objective we think longer term, which is how are we going to keep COVID down? Because we think that this virus is going to be with us for the long term. And there's some debate about how durable the initial vaccines are going to be. And we think that in some people, these vaccines are likely to be fairly durable. Protection ought to last for years. But in other people, the vaccine response, the immunity, may not be as durable, and it's going to be a good idea to boost them, maybe annually. And if we think about how we use vaccines now, there's a vaccine called MMR that protects people against mumps, measles, and rubella. And it turns out that you do need to boost people occasionally against their protection against measles and against mumps, but you just use MMR for that, which means that the R portion of it, rubella, is basically like a extra freebie that you get. You get an extra booster of rubella when you're really trying to boost for measles or mumps. And so as we think about how in the future people may or may not need boosting for COVID, but why not include it with your flu vaccine that hopefully people will be getting annually and more people will be getting to protect against COVID-like symptoms, right? Nobody's going to want to, be sitting next to somebody sneezing or spiking a fever in the future because they're not going to think flu, they're going to think COVID. And so, as you get higher uptake of flu vaccines, it seems to make sense to include an antigen in those vaccines that will protect people against SARS-2 as well. And for some people, that'll be a really useful booster shot. And for other people, it'll be just an extra freebie. It doesn't hurt them. It might just help, right? And so who has the ability to create such a combination flu plus SARS-2 vaccine? That's a question that we had on our mind as we went searching for the best vaccine technologies for the longer run. And we'll walk you through some data slides later that will show you that it's actually a limited number of companies that have that capability to make a combination vaccine. And they are using some of the more traditional technologies, not some of the cooler new ones that we've been learning about, like mRNA and adenovirus. So let's proceed through the tour now. So our map is structured with vaccines on the left, to prevent infection, and treatments on the right side, looking at the treatment of people who have gotten infected. So over here, we're going to focus on validated vaccine approaches first before we go into some of the newer methods. And you'll see here, validated kicks up to proven and widely used vaccine approaches that are injected. And by the way, our hope is that these tours teach you how to use this map, make it familiar to you, and you can keep coming back and examine it on your own time, show your peers, and ultimately we're disseminating knowledge of how to use this tool to let people be more independent of us. Within the proven vaccines, you've got these subunit vaccines over here that use basically protein, typically spike protein, that you inject into a person together with an adjuvant, which is an immune stimulating agent, in order to show the immune system what it needs to develop antibodies against. And you can either use full-length spike protein, it shows the whole structure of the spike protein, or just a portion of the spike protein. Oftentimes the most relevant one is the receptor binding domain, the portion of spike that actually binds to the ACE2 receptor on cells and that you would want antibodies to interfere with. So over here in the full length, you've got some old school technologies, virus-like particles that are have been used for other types of vaccines. But what you see here is a lot of companies that don't really have, well, they don't have CEPI funding, they don't have BARDA funding. They seem undercapitalized, at least at the moment. We would argue that they're a bit late to the game for now. And then you've got this mainstream player, Novavax, that has CEPI and BARDA and Operation Warp Speed funding at this point, and they're gearing up to make a lot of doses. We're definitely going to be talking more about Novavax, and full disclosure, RA Capital has financed Novavax. We have a financial interest in this company. So the thing that stood out to us, by the way, about Novavax was actually that they do have a flu vaccine in development as well. And we got data on that just as COVID was hitting. And that's part of what made us realize we shouldn't stop thinking about flu just because we're all focused on COVID. So the other companies that are working on protein vaccines are working on these truncated spike proteins, so portions of spike. And again, here you've got a lot of smaller companies and academic groups. We think they're mostly far behind and less likely to play a role in the response to the pandemic. Then you've got these larger companies. In particular, I would draw your attention here to Sanofi, which is a really established vaccine player. Like, they're one of the world's largest, and they generate vaccines every year. They really know the vaccine business, and they are behind others, but once they get going, once they come to market, they ought to have quite large scale production. So we don't have any clinical data to share for this Sanofi vaccine, but we're optimistic that whatever they make ought to be credible. And then finally, in the tried and true category, you've also got whole virus vaccines, where you just grow up a bunch of SARS-2, you kill it, and then you inject that, possibly with an adjuvant, into people, and they can develop immunity, not just against the spike protein, but against other proteins that are on the surface of the virus particle. And there's some rationale for thinking that might be a good set of programs to back, just in case we might be missing something, and vaccinating people with spike alone isn't enough. I think it clinical data to say that odds are vaccinating people with just the spike protein is going to be enough. So I'm less worried about these programs not having gotten all that much investment. But I think in the future, as we think about being prepared for response against possible pandemic viruses, I think we really ought to invest more into the whole inactivated virus platform. So then moving down to the lower left zone, we've vaccine approaches. And that's complex down below. And then up here, we're going to first talk about the less complex, potentially safer and more likely to work vaccines. And then some of the famous, often talked about adenoviral vaccines that are injected, and particularly J&J's Ad26 vector and AstraZeneca's ChAdOx1 vector. If anybody's heard of the Russian vaccine, you can pretty much think about it as 2 doses of this kind of vaccine, one of them being very much like Ad26, and another one being very much like CanSino's Ad5. In fact, this is a Russian company, Gamaleya, here that I'm talking about. They actually pulled an interesting trick. They declared that they got approval, for their vaccine in Russia, but in fact, they basically completed phase 1 trials. They haven't scaled up manufacturing, and so what they functionally approved is access to a vaccine that they haven't produced yet. So, they're only just embarking on their phase 3 trials. They won't start production until September. So if that's the basis for approval, you could argue that AstraZeneca, Moderna, BioNTech, and even Novavax could be declaring themselves as having approved vaccines. They just don't have any doses to offer anybody. So, I think it's more exercise in semantics, what we heard out of Russia for now. So here with these adenoviral vaccines, one of the limitations of these vaccines is that they develop immunity against the actual viral capsid that delivers the genetic code for the spike protein. So these vaccines can't really be redosed frequently. They will work less and less well. You inject them, some will reject the the actual adenovirus. So they're good for the initial pandemic response, but we likely to play a role in future booster shots. The other kinds of novel vaccines that we have in development are vaccines, and in particular mRNA vaccines we've heard a fair bit about. And there are also DNA vaccines. At this point, they are less likely to be effective. There's not significant investment in scaling vaccines. So we would draw your attention to the mRNA vaccines. And there are a lot of mRNA vaccines. The dominant players are Moderna, BioNTech partnered with Pfizer, and to some lesser extent here, though Translate Bio is partnered. We may see more data coming out of mRNA vaccine companies, but there's a lot of data now from Moderna and BioNTech, and we're going to examine. And the key thing to know about mRNA is that mRNA both encodes the antigen, the spike protein, itself immunogenic, reactogenic, it's the adjuvant. It really irritates the immune system and draws attention to that zone where you got the injection. And so if you try to give more mRNA for the antigen, but you're also using the immune system, stimulating it more and causing more side effects. So we're going to examine this of vaccines, and you'll see that this modality is turning out to be one of the more painful to tolerate vaccine. And then you a set of more complicated novel modalities that include replicating viruses like flu and horsepox and BSV. And these are likely to play a significant role. You can get a vaccine that isn't smallpox virus, that doesn't have an associated arthritis and replicating in joint you have here with VSV when it was used for an Ebola vaccine. I think people will probably prefer to avoid these if they can. But we'll keep an open mind. Certainly it's always interesting when a company as credible as Merck gets involved in a vaccine, and Merck has a measles vector and a VSV vector, and this one's even got Operation Warp Speed funding. So when the data come out here, if the data are compelling, we'll probably end up rejiggering this map and VSV up in the more credible zone. And then we track the status of these vaccine programs, when they first start in the clinic, when they're likely to get approved, and how many courses of vaccines each company is likely to produce in each period. And so when you sum all these up, all these vaccines from all these different companies, in theory, you could have as many as 741 million courses of vaccine. A course, in the case of a 2-dose vaccine, means 2 doses. So, a lot of these vaccines are 2-dose vaccines. So you're really talking about almost 1.5 billion doses of vaccine made by the end of this year. And that's all over the world. And the trouble is that realistically, all these vaccines aren't going to make it. We need a whole lot more than 741 million courses anyway. We need to vaccinate the world. And so we're going to be vaccinating the world all the way through 2021. And as these vaccine programs, reveal themselves to be not good enough, we're going to strip away some of these numbers, and we'll find out just how far into 2022 we're going to need to be manufacturing and vaccinating people. So we got to develop some pretty smart algorithms for figuring out how to deploy the vaccine doses that we have. And it makes sense that the US has engaged the US military in Operation Warp Speed in order to think about rapid, intelligent deployment of vaccine doses all over the country. So let's take a look at some of the data that's emerged from those companies that have generated clinical data. It's not all of them. We don't include groups like CanSino, but CanSino has a vaccine that's similar to AstraZeneca's, so that'll stand in for adenoviral vaccines. But first, let's talk about understanding how well a vaccine actually works. What are you looking for? Well, you want a vaccine to generate neutralizing antibodies, but how much? And the way to figure out how much is to take a look at, well, what are the neutralizing antibody levels in people who have recovered from infection? And it turns out that you have a range of those titers. You can have a huge amount of antibody in a person who's recovered from a serious infection. You can have, this amount, about 837 here, to 870 from people who had moderate infection. Maybe they didn't need to be put on a ventilator or have respiratory support, but they might have been hospitalized. And then these might be people who are asymptomatic or have mild infection. So you have this zone and you feel like, all right, well, if I'm getting antibodies somewhere in this zone, then I'm feeling like probably my vaccine is simulating the effects of an infection on training the immune system. But you also have to take a look at how are you measuring these antibodies. It turns out that there are different assays, different tests you can do. And the analogy I would draw is measuring the speed of a car. If you're measuring the speed of a car using a stringent assay, you would use miles per hour. If you're using one that's less stringent, in this case, how much antibodies does it take to only reduce the amount of cell infections that a virus can cause by 50% versus fully protecting the cells, protect them 100%, then it's kind of like measuring speed of a car using kilometers an hour. So a car going the same speed will appear to be going slower if you measure in miles per hour than if you're measuring in kilometers an hour. So the less stringent the assay is, the better it makes your antibodies look. That doesn't mean that your antibody levels are on an absolute basis better. It's just different ways of measuring. So ultimately, when we then look at the results that these various companies generate in their clinical studies, what you can see here is that you've got, in some cases, data on how much antibody the first dose, the prime dose of a vaccine generated. So in this case, neutralizing antibodies reached 100. Second dose reached 3,900. And they included a control where they used their assay, in this case, a very stringent microneutralization 100% assay, to see, well, what does their assay say about the level of antibody in some control samples of people who have recovered from COVID? And you see that's 983. And so after their second dose, they're about 4 times better than that control. And in the case of Moderna and BioNTech actually has 2 studies here, their vaccine, in the case of Moderna, they don't report how much the antibodies got to after 1 dose, but after 2 doses, it gets up into the mid-hundreds here, which is a lot less than 3,900, but you also see that the controls they looked at, were lower. So here, this looks about 4-fold better. So you might think, well, okay, that's 4-fold, this is 4-fold, so does that mean that they're the same? Well, again, you have to ask, what controls did they pick? Could it be that these are, there's only 3 patients here, maybe they picked patients that were comparatively mild, right? And had relatively low antibodies. So it is curious that these other clinical studies had control data that looks so low. But it is reassuring to know that their vaccines at least did much better than that, right? And you see some antibodies being generated after the first dose. By the way, that's important to generate some antibodies after a first dose, because when you're vaccinating people and they get the first dose, especially if it's painful, what if they don't come back? You want to know that they're at least a little bit protected. I feel like I probably have bad internet here and I might be juddery. So apologies for that. And then AstraZeneca, they have a control here. It has really low antibodies in the control. And their vaccine generated neutralizing antibody titers even less than that. That's pretty disappointing, actually. So AstraZeneca is in the lead. They're running some large, find out if it's protective. But if they're not protective, then it might be because they're just not generating enough neutralizing antibody. And we'll be hopeful that these other vaccines that are right behind AstraZeneca are going to be better. If they are protective, if it turns out that this can really cut the amount of infection from COVID, from SARS-2 by more than 50%, we're going to feel a lot better about these vaccines, because we'll know that if AstraZeneca is protective, then probably these are great. So the way to understand this whole space is, what we're trying to get to is that you want as high neutralizing titers as possible using the most stringent assay, and you'll see shortly, not everybody uses the same assay, and versus the most robust control. So you want the highest neutralizing titers versus stringent assay, and here Novavax has the most stringent and AstraZeneca has the most stringent. Moderna's is moderately stringent, and versus the most robust control. And Novavax has the most robust control. These others, maybe they're good controls, or maybe they're representative of milder disease. It's hard to know. But when you look at this, what you realize is these are the data that show you the highest neutralizing titers using the most stringent assay. So this is miles per hour. This other stuff here is kilometers an hour. And versus the most robust control. So this is one of the reasons, actually I would say I've just summarized the reason why a lot of people, upon looking at the clinical data, are now pointing to this data set, the Novavax data set, as the best so far of the bunch. So that's maybe not as important as asking the question, what is good enough? And we won't know that till we get the result of larger protection studies, the kinds of studies that have like 30,000 patients. And if it turns out AstraZeneca is good enough, odds are all the rest of this is too, and that'll be great. We'll be able to end the pandemic sooner. If this isn't good enough, the next question is going to be, is this good enough? And I actually think it likely is good enough. But if it's not, well, then you'll know that even better is coming after that. There's also some interest in the question of what kind of T cell responses these different vaccines generate. Now, the trouble with T cell responses is, while it is a branch of the immunity that's definitely involved in protecting people against infection, we actually don't know its significance when it comes to vaccines. We know that T cells play an important role after you've recovered from an infection, but we don't know what the role is of T cells if you got a T cell response to a vaccine. What we do know is, like, having some is better than having none, and more is better. So here again, Novavax reports in their studies, these levels, and other companies have lower. These are not head-to-head, so there may be, differences in the stringency of a good enough understanding of all these assays to tell you what's more and what's less stringent. So this is just, to us, it's observational and directionally interesting. So let's take a look at safety. Now, these data we've actually got embedded together with the data we showed you on titers on the map itself. So you'll be able to come back to the map and review these data in some depth. And in order to put the tolerability data for these vaccines into context, we include here some influenza vaccines, so 2 of Sanofi's flu vaccines, and also Novavax's flu phase 1 data, as well as, for really good comparison's sake, Shingrix, which is an incredibly effective vaccine against shingles. It's also the most painful and of all of the commercially available vaccines. So people still get it. Millions of people get this vaccine. And so that tells you that however painful it is, it's tolerable. But you would not enjoy taking vaccines that are as painful as this or more painful. We also include placebo arms, which will give you a sense that people can report feeling pain just from the injection, and they can even report feeling things like fever just from a placebo injection. In this case, this little red sliver there shows a placebo patient reporting severe fever, right? So not every side effect that you see is necessarily due to a vaccine. We also break out the safety data based on the first dose of these vaccines and the second. Now, flu vaccines, you only get 1 dose, but in the case of shingles, you get 2. And in the case of COVID vaccines, in most cases, you're going to get 2. And what you'll see is that the tolerability of these vaccines gets worse with the second dose, typically. Now, we cover a whole set of these side effects. You can examine them more up close on the map. I'm going to actually show you a different representation so you can take in the totality of the data using this color scheme. So we'll show you in green which of the vaccines are, and the side effects are basically undifferentiated from placebo. So you can see here, this flu vaccine, sure, it causes some headache, but no more than the placebo does. And so we would say that's basically placebo-like tolerability. Where you see yellow, we're saying, well, that's typical for a flu vaccine. So it's really not worse than flu vaccines. And where you see orange, you'll see that's what Shingrix is like. And in some cases, Shingrix is no different than placebo. In other cases, no different than flu. But other times, it really defines the severity of the space. So here, for local pain with some of these severe cases, Shingrix is distinctly more painful. So let's take a look at some of the COVID vaccines. You see AstraZeneca here. It's got local pain akin to the shingles vaccine. Swelling is like placebo, but fever, well, that's off the charts. So that's worse than Shingrix. Worse headache. For the mRNA vaccines, their pain is worse than Shingrix. Headache worse than Shingrix in this case. So the mRNA vaccines are looking to be, on their first shot, worse than the most intolerable commercially available vaccine. Hopefully in phase 3 trials, we'll discover that somehow it's better, but we got to prepare ourselves for the possibility that these are going to be unpleasant vaccines. So, and then Novavax's first dose, was milder. Here you see local pain akin to flu vaccines, placebo-like swelling, fever, headache, and then on the fatigue, that's where we scored that as ultimately even worse than potentially Shingrix. So some muscle pain here. Now on the second dose, Shingrix, for example, Shingrix fatigue is, much worse. You get chills from that, even some severe chills. And you look and you see like AstraZeneca is probably about comparable on the second dose to the first dose, but the mRNA vaccines are even worse. The pain is greater, the fever. You look at these fevers, you're going to be seeing people spiking fevers after the second dose. That's not common for vaccines. And Novavax is also on the second dose a bit more difficult to tolerate than the first dose. You got some red here in the headache and fatigue zone, but still a bit milder than mRNA. So the bottom line is COVID vaccines are not going to be fun. In some cases, not after the first dose, and in most cases, not after the second dose. So we got to be prepared to coach the public through that. And as we think about, yeah, but what about the seasonal booster shots? Well, we already talked about how adenoviral vaccines can't really be redosed because you develop immunity and they work less and less well, as you give them. They fail to deliver their instructions to the cells because the immune system just rejects the adenoviral vector. mRNA vaccines, pretty painful off, just the first dose. So getting that every year, and also, creating a pentavalent vaccine that has protection against 4 flu strains as well as COVID, you'd have to give 5 times as much mRNA. And that's, we think, going to be even more difficult to tolerate than you see. And so you look at the Novavax profile, and it looks more like something that you could imagine giving each year, right? And it's possible for a booster, they might give less of the adjuvant that they use, matrix M, and make it a bit more tolerable. And thereby create a more tolerable flu-like pentavalent vaccine, right? And since you're only going to give 1 annual booster, you don't have to think about the tolerability of a here. So that's how we're thinking about these vaccines at the moment. Tolerability, the first dose is important since you want people to come back for the second dose to get really well protected against COVID. And you want people to come back for booster shots. We'll all likely be getting with some regularity, probably together with flu shots. All right. So I will now pass it on to my colleagues for you to hear about the therapeutic space and the diagnostic space. Thank you.
Thanks, Peter. Great. I'm just going to share my screen now. All right. Can everyone see the screen? Yep, hopefully. All right. Hi, everyone. My name's Alyssa. I'm an associate here at RA. And thanks so much for going through in great detail about vaccines, Peter. I think everyone is really looking forward to understanding how they're going to help us, work through this pandemic and also prevent future waves. But really, in the meantime, we're going to need therapeutics that work because vaccines are not yet available. We don't know how effective they'll be, and some people can't take them and some people won't take them. So the right side of the map is really dedicated to outlining all of the therapeutics that are in development for COVID-19. So you can see we've kind of broken it down into 3 separate strategies, 2 of which we'll be talking about today. So one, which you're probably quite familiar with, is targeting the virus itself. There are lots of antivirals out there for other viral infections. You might be familiar with Tamiflu if you've ever had influenza. And these are drugs that actually prevent the virus from replicating within your own cells and can clear the infection. Now, we'll also talk about some strategies that I think are a little more unique to a COVID-19 infection in the sense that some patients who are infected with COVID-19 end up having a very aberrant immune response. That is, their immune system sort of goes into haywire. And so trying to dampen down that immune response can actually prevent some of the self-inflicted damage that you might experience if you kind of have that immune response due to COVID infection. And my colleague Ganesh is going to take you through some of those strategies. So the first drug that I'd like to sort of talk about is the antiviral remdesivir, which I think probably just about everyone has heard of now. So it's been approved for emergency use in patients. It can be found in this area on our map. And one thing that I'd like to note is that so remdesivir is an antiviral. It blocks the RNA polymerase that really copies the viral genome to make several other copies of itself. And so remdesivir was demonstrated in a clinical trial to show that basically it could reduce the time spent in hospital by about 30%. And this is actually quite meaningful when you take into consideration how crowded some of those hospitals are. I mean, I think everyone saw the news stories of New York back in March and April, and now what's going on in Florida and Texas and California. Some of these hospitals are really reaching capacity, and so if they can give patients remdesivir and help them get better quicker, that really opens up space for patients to be able to, for more room for new patients to be able to be treated. So remdesivir, it's approved for use, but it's not without limitations in the sense that basically remdesivir has to be given intravenously. And what that means is that someone needs to be in the hospital to receive it. And so generally it's being reserved for the most sick patients. Now, it's nothing unique about this mechanism of action that would make something that would need to be intravenously infused. It just has to do with the chemistry of remdesivir. And so what we've mapped on the map as well is really highlighting that while we have remdesivir available to us, a lot of other companies are actually trying to take that mechanism. So maybe either take remdesivir, deliver it differently, or take other drugs that block the RNA-dependent RNA polymerase that copies that viral genome that can be given orally. And so again, sort of the benefit there really is that these could potentially be given to a wider variety of patients, right? You don't have to be in the hospital. You don't have to have this IV infused into you over 5 days, but it could still potentially reduce sort of the duration of symptoms that you may be experiencing. Again, kind of similar to the example I gave earlier, if anyone's ever had influenza, taking something like Tamiflu can actually make you feel better faster. So there are a variety of companies that are working on this. Ridgeback and Atea, I think, are two that have been in the news a little bit lately. But we're really looking forward to seeing sort of the data come out of these approaches, because one, it'll allow for a larger supply of hopefully drugs that are effective in reducing sort of that viral replication within a person. And two, it will expand the time at which someone could receive such a drug. So we will, we are tracking that, and as soon as we get the data, we will update the map to help teach what approaches work and maybe if any don't work. So in addition to sort of blocking the viral replication, so here, we talk about taking remdesivir, which blocks viral replication. There are also approaches that can block or prevent the viral entry into cells. So maybe not necessarily preventing, infection once the virus gets into the cell, but actually creating almost like a physical barrier to prevent that virus from ever even making it into the cell. And so these are generally antibody approaches in nature. So it's quite similar actually to how Peter was describing earlier. When you get a vaccine, your body mounts an antibody response and that can block the infection. Well, in these approaches, we're actually creating those antibodies that we know should block the infection and then infusing them into patients. And so for these prevention of viral entry, you can see there are a ton of different approaches that are utilizing sort of antibody-like approaches to prevent that infection. And one thing that I would just like to note is that, there is precedent for this from other viral infections. We know that there are antibodies that have been developed for Ebola, antibodies have been developed for influenza, but we also know that something called convalescent plasma can be effective in treating and preventing, viral infection for patients who are infected. And so what convalescent plasma is essentially taking plasma from a patient who has been infected by the virus and then, has subsequently cleared the viral infection. So we know that they have a lot of antibodies, so like Peter was talking about earlier. But then actually taking that plasma and infusing it into a new patient to act as a therapeutic. And we've seen, there have been some, I would say, anecdotal stories where folks do believe that this has helped patients. We haven't seen any very robust clinical trials in COVID-19 for this approach, just because it was quite challenging in the beginning when everything was, when all the doctors and all the hospitals were having quite a difficult time trying to treat. I think some of the protocols designed to test this hypothesis were maybe not quite as robust, but we do know from other diseases that convalescent plasma works, and that helps us give us a little more confidence in the idea of using antibodies to prevent viral infection. Now, one of the challenges with convalescent plasma, like I said, is you're actually getting it from human donors, and so the amount of convalescent plasma that you can actually get from one donor is not quite that much. And so what the pharmaceutical companies have done is really understood, well, can we pick out what antibodies we think are the most important based on, the spike protein and other portions of the virus that they think blocking should limit its infection of cells? And can we make those proteins recombinantly and then dose those? And so here, kind of on the map, we talk about monoclonal antibodies that are against the spike protein. And there are a lot of different approaches. So you can either think about advancing a single antibody, which there are plenty of folks who are working on that, or you could also potentially make a combination of antibodies. And so this would be a little bit more like convalescent plasma, and it also could potentially prevent any mutant or any resistance to the antibodies. And so I think we're very interested in a lot of these approaches and really look forward to seeing what the data look like, because not only can these be given to patients who have the disease, but we could also envision a situation where maybe high-risk healthcare workers could receive antibodies that are for the virus, and that could actually work in a prophylactic setting. So again, we'll be monitoring these trials and updating as we see data, but I think this just, again, really opens up the armamentarium of what we could potentially utilize to help protect patients and help hopefully get patients who are very sick better quicker. And so now I'm going to actually hand it over to my colleague Ganesh, and she is going to talk about sort of some of the immune strategies that folks are taking for COVID-19.
Thank you, Alyssa. Let me share my screen as well. Sorry that it takes some time. So I will try to walk you through on the right side below section about the therapeutics that would be useful for the critical and severely ill COVID-19 patients. Did my screen share work?
It looks like it's still thinking.
Yeah, sorry for that.
Do you want me to share mine and I can advance for you, Ganesh?
Did it work now? If not, please do that.
Okay, here, can you unshare?
Okay, let me stop sharing. Sorry for this difficulty.
All right, sorry, let me get it back up. Okay.
As my colleague Alyssa already elaborated on, that our immune system is actually our biggest weapon to fight the virus. However, it gets overactivated. So since it gets overactivated and it gets high-wired, 6% of the symptomatic patients can become critically ill and they develop acute respiratory distress syndrome. Thank you very much, Alyssa.
All right.
And when we go to the central node, in the red section, we see the potential treatments for those patients. So to achieve a prevention on onset of cytokine storm, if you can move one step ahead, Alyssa, we try to divide the map into two different sections where we were looking for inhibition of specific cytokines versus targeting the immune system in broad way. Till now, the only success in the red section has been a generic steroid called dexamethasone, which has been shown in a clinical trial to reduce mortality. Given that we observed in US more than 170,000 deaths, dexamethasone showed in a clinical trial that it can reduce mortality in 35%, by 35% in the ventilated patients, which are the most severest population, and in the 20% in the patients who require oxygen support. Beyond this great success, a lot of people were very excited and hopeful on targeting specific cytokines will also lead successful outcomes. However, targeting specifically immune system via the targeting IL-6 cytokine, which is one of the major cytokines which gets over-regulated in ARDS, acute respiratory distress syndrome patients, has unfortunately not worked so far. Kezara, Serlimab from Regeneron and Sanofi, and also recently last month, we heard from Roche's trial, Actemra tocilizumab, both has been failed. Roche is still trying to figure out whether a combination of tocilizumab with remdesivir can improve clinical outcome or at least reduce mortality, which we are looking forward to hear in the next quarter. However, as a conclusion from this drug class, that it doesn't look good that hitting one major cytokine may not be good enough for the COVID-19 patients. Beyond all these disappointments, we got some hope from another approach, which is more immunomodulatory, where it can target several cytokines at the same time. This came out from anti-CD6 monoclonal antibody. If we can move to the next section, Alyssa, thank you. You would see that Equilium with bioconitolizumab showed actually promising results and received emergency use authorization in India. However, this is a very small clinical trial, only in 30 patients, and we are looking forward to hear a global study outcome for this drug. And hopefully, if they show clinical benefit, it can be another weapon that we will have in our toolbox to fight against COVID-19. So beyond all these, similar to the vaccine section, we are keeping a track of all the drugs that are gonna have a readout for the therapeutic section, including the part that my colleague Alyssa covered, which has been done together with the orange section and the red section for the mild patients, and for the critically and severe COVID-19 patients, this adds up to almost 250 therapeutics in the pipeline. So what we hope is beyond to have another drug, to have beyond remdesivir and dexamethasone, so that we can combine with the new drugs and get even additional benefit for COVID-19 patients. We need extremely distinct mechanism of actions which would enable that we can combine those. So if it would be just another steroid, it will be too much immunosuppression for the COVID-19 patients. But if it's a unique mechanism of action, we would aim a unique, newer tool set. So we are looking forward to hear from this quarter and next quarter another set of drugs, outcomes from the clinical trials, and we are hopeful, and we are looking forward to update you also in the next MAP update. With those words, I want to hand it to Nate, who will cover the diagnostic map and tell you and share with you all the insights regarding. Thank you.
Thank you, Ganesh. So I'm sure you've heard about testing COVID diagnostics in the news and the failures that we've had in that space. So we launched a, I can take it, Alyssa, I think. We launched a COVID-Dx map to help explain some of the trade-offs and uncertainties and opportunities in that space. Just a second. And so, like all of our maps, we start in the center with the questions. Can you see my screen? All right. And there's two main questions I'm going to focus on here for COVID diagnostics. The first is what most people have heard about. Who has COVID? And these are the molecular tests. And the problem here has really been about scale. The tests are good. We just don't have enough of them. On the other side, we can scale who is already immune to COVID, and we use the surrogate of who has had COVID here. The question is more about accuracy. And we now have accurate tests, but certainly at first that wasn't the question at all. But now the question moves to what is the right surrogate? And we're starting to get evidence on what level of antibodies are needed in order to confer real protection. And we'll know a lot more when the vaccine trials read out and we understand more about that. So we're running around 800,000 tests a day. These are molecular tests. And the reason that we have had trouble scaling is that these tests are run on equipment that is typically used for HIV, HPV, gonorrhea screening, and 800,000 tests a day is far more than we've ever tried to run in total for this type of test. And so we're running into bottlenecks on extraction and originally swabs. But now we're really running into the bottlenecks on how much lab space do we have? How many people are trained and able to do this? And you know, there's multiple approaches about how many tests a day we need, but I think we need to be thinking about, you know, orders of magnitude more testing than we have today in order to be catching all cases and try to mitigate the pandemic that way. And that's part of the rationale behind our launch of COVID Apollo project. But here we can see if we move away from this paradigm of thinking about tests as diagnostics, as trying to pick up, you have a patient you suspect has a disease, do they really have this disease? And move it towards, we're going to test a patient who has no evidence of disease just to see if they have it, because so many of people who contract COVID-19 and spread COVID-19 do not actually show any symptoms and would never show up to the hospital. And the amounts, you know, the number of patients has certainly varied, but it's probably around 40% of patients never show symptoms or show symptoms that are so small that they don't realize they've had it. And so at the top is sort of the current approach that we've taken so far. At best, some nursing homes are doing weekly screening with a very high sensitivity assay. But the problem is that this really only can possibly catch 40% of infectious days, and we don't have enough of these tests to run anyway. And so as we move down this, this chart, we see that even tests with quite poor sensitivity, and even 40% clinical sensitivity, which would never sort of pass the bar for a diagnostic, can really have a meaningful impact on the disease if they are rapid turnaround and can be run often. And so this brings us to the COVID Apollo criteria of, you know, rapid, fast, and less than the price of coffee, so that people will actually use them. So when we, you know, as I said, the problem of who has COVID testing is scale and turnaround time. And so a central lab test that takes 48 hours to come back is really not that useful for screening, because you lose those 2 days of potentially a patient going out and going to church or going to a ball game and spreading the disease to hundreds of others. And so on the other side, on the serology or antibody testing side, the challenge has been accuracy. And while, you know, a 95% accurate test sounds pretty good, if you have a 4% population prevalence. If you're testing a group of people and only 4% of them have actually had COVID and your test has 95% specificity, and you're one of those people and you get your results and it comes back positive, you actually only have a 40% chance of really having had COVID, because the population prevalence is quite low. Because if you run 100 tests, 5 of them will come back falsely positive. And that is really dangerous for these tests that are meant as a surrogate for am I immune? Which is why the importance of these very, very accurate tests with greater than 99% specificity, which you can see here with a test like that, such as the Roche or Abbott or some of these other tests, at a 4% population prevalence, if you get a positive test, you're 94% likely to have actually had COVID and potentially be immune. And so while we've developed these tests, now it comes to the challenge, which is to which of these people are really immune. And we're starting to see that there's early data saying that for the really good tests, it's potentially 80% of people are really immune, have neutralizing titers. And there's some really, really early evidence with just a few people indicating that even those low titers may be protective, but there's still a lot of work to be done here and a lot more that we need to know. That's a brief overview of COVID diagnostics. I'm going to pass it back to Peter to close.
Thanks very much, Nate. As far as diagnostics go, I don't know about if anybody else has had this dilemma, but we at RE Capital are trying to figure out how can we hang out again. I mean, we just sort of miss each other. It would be nice to get together and have a drink. And we've been looking into some of the testing services that might allow you to send they'll send you a test. You would perform it on yourself, and then you send it back. So like the Pixel test that LabCorp offers. And these tests rely on a nasal swab, which made us think, well, is that less accurate, than nasal pharyngeal swab? We thought that it would be. And the cool answer was, well, sort of yes, sort of they are less accurate. On the other hand, if they miss something, odds are it's because the amount of virus that you have is so low that you're probably not infectious. So the argument is you're not missing anything important. So that's somewhat reassuring, and we're going to be looking for confirmation that that's really true. We looked into antigen tests. We have not found anybody that will allow us to do a send away rapid antigen test that we would consider to be accurate. They have antigen tests, for example, at the White House. So if you got that kind of pull, maybe you can get those machines and do rapid antigen. For the rest of us, the most convenient way to have something that's just shipped to your house that you can do is one of these nasal swab PCR tests that send away, and it takes several days. But you could imagine having everybody on your team, or anybody in your family get this, and haven't engaged in any kind of risky behavior in the few days prior, then if they had an infection, if they had gotten infected, let's say 3 or 5 days prior, they ought to have high enough levels of virus that the test ought to detect it. And then as they wait for the result, they got to stay social distance and safe. And then you can get together. If you had an entire family tested, then if everybody in the household was negative, that you feel better about everyone in the household actually being negative, as opposed to testing only one person and maybe it's a false negative. Right. And so we then looked at the cost of it, and it's a few $100. And you have to, by the way, tell them that you have COVID symptoms. So maybe you've sneezed or you've coughed, but if you don't have symptoms, they won't even send you this test. So we're not at a point yet where people can do this kind of like broad testing before they get together, just as a precaution, and that's a problem. It's a shame. And if we tried to do this for our firm routinely, everybody says they've got a cough, you get tested once a week just so that we're free to get together whenever we like. The costs end up being, for a firm of our size, like 90 people, really off the charts. So, we even looked into, should we get our own equipment and do this? It's a shame that we can't even create a pocket of safety like this if we want to. Now, if you're a Major League Baseball team, I guess in theory you could, but that's because they're trying to provide entertainment for the whole country. And I guess nobody considers RA Capital entertaining enough just yet, to give us access to that kind of rapid turnaround. So we continue to work on this with a very, sort of personal on the ground interest, like how would we actually solve this for our team? And if we crack that, we'll share with you how we did it, and you might consider doing it for yourselves. But if you come up with a solution that would allow your company or your group to easily be able to get together and reassure one another that you're negative, please let us know. If you're from the White House, please don't rub in how awesome your testing capabilities are. We'd love to have them. All right, thank you, until next time. Thanks again, team.
Thank you, and we'll leave the question and answer up for about 10 minutes in the event that you'd like to go through them. Thanks all.